chip that I'm holding in my hand
Yeah.
and read my DNA, sense the process of clipping the DNA and programming it.
Yes.
This is the gateway to digitize the body.
Yes, it's a gateway to
Wow.
biology. I could see a future where we human collaborate with nature and biology, and we create technologies that is an integration of human-made technology and nature-made technology that leads to really powerful technologies.
Why do you think that we're all robots?
Interesting question.
When you say robots on enzymes, what is it that these enzymes do in our body?
These are the ones that are responsible to, cutting and breaking down the food to these small particles. Then they go to your bloodstream.
And we can control those enzymes.
I mean, I remember when I first presented that project to my colleagues, everyone laughed at me. What we also realized, so that sometimes when you invent something, you actually don't know where the application would be until you start doing it.
What happened to the insulin? Were you able to deliver insulin?
Well,
What can be done in the dental world? Why do you think that we're all robots?
interesting question. So I think we're all made of micro robots. These are all the little machineries within our body that are responsible for some sort of function. So our tissue are sort of a larger scale, robots, and then you go down more in a scale, and then you get to, cells and molecules within the cells. This is from your nucleus to your mitochondria to your the enzymes that are produced within your cells, and each of them have this very, very specific function. They carry things around, they cut things, they assemble things. So it's really a whole assembly processes and fabrication, processes that are going on inside our bodies that we're just recently starting to learn about them. So that's why I think we are a big robot, consists of many, many small robots down to nano-size creatures that are working within our bodies.
And your job is to control that robot with machinery?
Yeah. So my job is actually try to understand what they do and see if we can use their function for something that is beneficial for us. Of course, they're doing something that are beneficial for us. But when we talk about these little micro robots, we're not only talking about things within our body. We're talking about things within plants, within other organism, insects. Can we get the best machinery out of every creature, and then use it for something else?
Can we open it a little bit more? When you say robots on enzymes, and what is it that these enzymes do in our body?
Yeah. So I'll give you a simple example. When you eat food, there are enzymes within your gut that are responsible to cut down these foods that you're eating, sugars, fatty acids, and make it small enough so they're able to get to your blood, to your circulation system. This is how you're actually feeding your organs and tissue with these small molecules like glucose. Because you're not eating glucose in its pure format. Glucose is sugar that you ingest as a part of your food. And now the enzymes function within your body. These are the ones that are responsible to cutting and breaking down the food to these small particles that are then they go to your bloodstream, and then they are transferred to your tissue.
And we can control those enzymes. This is scientific, proven.
Yeah, I mean, now, so I have to take you a bit back because...
Please.
Yeah, so we're just recently starting to have access to these enzymes and understanding their function. When you think back, the invention of telescope, which was sort of close to when microscope was invented, this was basically our first glance. They gave us the first glance of a cell. And then we realized that there are these cells within our body that we didn't know about. Then, with invention of Marie Curie, she invented, this agent that we were able to. You guys do X-ray all the time, right?
Right.
We were able, with understanding those, wavelengths and how we can penetrate the cells. Then we started seeing this organism within the cells, which was like the mitochondria, the nucleus, and then the sequencing technology came about, where we were able to sequence the first DNA in 2000.
What year was this?
In 2000.
2000.
Yeah, that's the one. The first DNA was the first human DNA was fully sequenced, and then we understood that wow, these proteins that we see in our body, this enzyme, they're all, basically have been produced from this DNA code. So the DNA is basically just the software within our cells that then creates that, enzymes, proteins. Then, as our technology started advancing, the advancement of technology resulted in us understanding more of our biology.
Mm-hmm.
So now we know, for example, what enzymes are doing, what are their functions. Some of this enzyme, are responsible to take the DNA codes, take it to the nucleus, make a protein out of it. Then the protein comes out, and there are some enzymes that are responsible to making sure that that protein is designed in a certain way. And it's just fascinating. And we're just at the beginning.
Beginning of it.
Yeah, and as we're understanding the function of this enzyme, I am looking to see what do they do. Can I actually use them for something else? Can I capture them for something else? And it's really, some people may think, okay, how much enzyme you can take, from a cell, but that's not even an issue anymore because our technology have advanced so much that we can make this enzyme synthetically. There's a whole area of we call synthetic biology.
Mm-hmm.
Where we can make proteins, we can make this enzyme all in a synthetic way. So there is a scalability a potential for all of this enzyme. So I could see a future where we human collaborate with nature and biology, and we create, technologies that is an integration of human-made technology and nature-made technology that leads to really powerful, technologies.
And that's part of your research, essentially. Yes. How did you get interested in this field?
Yeah.
What's your education background?
So that's a very interesting question. I think my path to what I'm doing right now was sort of never defined. And it was always just answering a curious question. And some of those answers led to a product, a good technology, and some of them led to nothing, basically. So my background is electrical engineering.
Wow.
As you can imagine, I'm Persian, and if you're in Iran, it's like you're either an electrical engineer or a doctor.
Very true.
So when I came to US in 2002, I thought I had to be an electrical engineer, but I truly hated it. I hated making transistors and diodes and antennas, and but I was always good in my field, and I tried to be the top students. But I was really still looking, for something that I could connect with. I could see the impact of, because building transistors and diode, you can't use them right away to see what impact you're making. So I decided to go to medical school, biomedical sciences, and realized that being a doctor was not also for me because I felt like you're so consumed with the amount of work that you're going to do that it sort of takes you away from the daily science, the advanced science. So that's why I decided to do biomedical engineering for my PhD. And this is where I started trying to figure out how I could use my electrical engineering knowledge to bring some biomedical solution. Mm-hmm. So multiple projects here and there, but it was not until my postdoc at Berkeley where I gained, I think, enough intellectual bravery where I had no issue of coming up with a crazy idea and going after it, And once one of your crazy ideas work, then you become more brave to pursue the other ideas. So I think this is how I got to trying these things. And, the first technology I actually developed was an ingestible electric capsule that you would swallow and it would get to your gut, gets a signal as it passed the pyloric valve and then ejects the drug with high pressure. I remember when I first presented that project to my colleagues, everyone laughed at me because the way the pill was working, it sort of generates gas as it gets to the small intestine, and that gas will then pushes a piston, gets to 50 kilopascal, and then once it gets to 50 kilopascal, then the nozzles would break and eject the drug with high pressure, which allows the drug then to penetrate and gets to your...
I want to hear, how did you choose that process and project?
Your mentors they play a major role, right? So my mentor was a pure bioengineer. So that he used to make molecule drug delivery formulations. So he asked me, Kiana, make me something that can deliver insulin orally.
Oh, wow.
So
we can't deliver insulin orally.
No.
Why?
No, because there are multiple reasons. So insulin is a protein-based drug, right?
Uh-huh.
And then when you ingest proteins, your body is designed to
defeat it,
to cut it,
to
chop
break
it, right?
it down.
And
Yeah,
as with those enzymes,
enzymes.
Yes, those enzymes are in charge of cutting those proteins. So when you take an insulin, even if you protect it so it's not impacted by the acidic environment of the stomach, which sort of destroys everything, when it gets to the gut, then it's basically broken down by those enzymes. So you're not getting a lot of active drug in your
bloodstream,
blood. Yeah. The other problem is that with insulin, it's even more difficult because insulin has a very narrow therapeutic window. That means that you have to really deliver something in a range. You can't deliver below because it's not effective. You can't deliver over because then it has side effect. So that was a challenge. And right now we don't have any oral delivery of insulin. And when you think about it from a patient perspective, it's really not cool to constantly get injection and poke yourself with needles. And it's not only insulin. There's a lot of other diseases like psoriasis or people who have to take Humira. They constantly have to inject themselves. And we really wanted to deliver a method to sort of make this drug orally. And so, of course, I was given a challenge. So I had to think about how to solve it. Then I thought about the jet injectors for vaccination. I don't know if you remember them. I think it was in 1980s where we had this gun vaccination systems where it was basically a huge gun that you basically
big
vaccinate.
Metal ones, right?
Yes, you vaccinate, and then it was a high pressure gun that would basically eject the drug with very high pressure. But we're talking about 1,200 kilopascal.
Right.
So that's a very high.
100 kilopascal.
Yes, because you have many layers of cells within your skin that you have to penetrate before you get to the blood.
A patient swallows the capsule, which contains a dose of medication and is about the size of a multivitamin.
So it's like a multivitamin site goes down?
Yes.
And how do you know when to activate it?
So
how do you give it the signal?
So the capsule I designed was based on time. So it would activate after 2 hours. And our gastric transit time, that means the time that it takes for your, when you ingest something to get to the small intestine, for human is between 20 minutes to 2 hours. So I designed it to be 2 hours. However,
time-based.
It was time-based. But however, what BioAura did, BioAura actually licensed another technology from Medametrics. And Medametrics had a diode. So the diode constantly will shine light and receive lights back. So as the capsule passes the pyloric valve, you know exactly you're passing the small intestine, the stomach now. It's time to eject, right? So then that would, So they combine the two technologies together, and what you see now is a combination of the two technologies.
Okay, so that research was your postdoc.
Yes, and
how did it become a company? What's the story there?
So first of all, I mean, there was a lot of challenges with just testing the capsule because we first tested it in rabbits. And at the time, I didn't know the GI processes of rabbits.
Is it different than humans? I'm
yes,
sure it is.
very, because they're grass eaters, right? So you need more time to digest the food in the stomach. And so anyways, it took me 9 months to get the animal protocol approved in Berkeley because they're very adamant to make sure that you're treating the animal in a humane way.
It's Berkeley, so
yes, yeah.
No surprise.
Absolutely. So the first time when we gave the capsule to the rabbits, it was just a safety test. And me and the vet, we were supposed to make sure that the capsule would come out intact. So 24 hours later, I'm still poking the rabbit's poop, trying to find the capsule. Yeah, there was no, sign of the capsule. The vet was panicking. She's like, "Oh, probably it degraded." And of course, I 3D printed the capsule.
Okay.
Yeah. So, and I'm like, plastic cannot be degraded in 24 hours. It's impossible. But it was difficult.
Plastic, if we could degrade in 24 hours.
Exactly. But then come and explain that to the vet. So anyways, we ended up euthanizing the animal, looking all over the GI tract.
Like opening him up?
Yes, opening up, looking to see where the capsule have gone. Because
Oh,
then
right.
the second thing is that, the second thought was that did it rupture the intestine and went somewhere, or it's just stuck in the
Sure.
intestine. It was nowhere. And when we were getting really like frustrated, then we found out that the rabbit was holding the capsule in his cheek the whole time. Because
He didn't even swallow
he
it.
didn't even swallow it, because rabbits do not like to swallow big things. So the following experiment, we had to really gavage the capsule. And then the first experiment we did, we put some X-ray reagent to be able to take X-ray from the rabbits to make sure that the capsule was passing in a right way. So it was an
Wow.
interesting process. And
Fun research.
it was fun, but at the same time, when you're in it and you have to deliver and you have to publish,
Pressure.
there's a lot of pressure. But I think the learning from that was
And what do you
amazing.
measure? The blood to see if the drug
Yes,
is there? That was
exactly.
the response.
Exactly.
Amazing.
Yeah. We would take continuous blood samples to see how much drug has penetrated.
And then you started the company.
Yeah, I started the company, and because in Berkeley everyone was starting companies,
Yeah,
and
of course.
but I was so naive. I had no idea how to pitch to VCs. I would go with a couple of slides of data, all data, and I assumed that everyone is visionary, that they would understand the impact. And of course, I'm sure you
No.
know, that's not the case. So it was a very harsh process for me. I was actually pitching to a lot of people who Elizabeth Holmes was pitching to.
Wrong timing, probably.
Wrong
too.
timing, yeah. So-
What year was this?
So it was after.
After his conviction, or
No, it was around the time that people were starting to understand that something was wrong with Theranos. And
Oh, you got burned. Wow.
so I realized those years later, after the Bad Blood book came out. And I'm
Right.
like, oh, this guy was treating me this way because of his bad experiences. So I started,
Biases, wow.
understanding. But anyway, so we women entrepreneur were not received well by VCs. Let me put it this way, especially the Silicon Valley. So I think it was, I could not raise money. I could not raise enough money to be able to take this to pig studies. And at the time I had the option to license my own technology from Berkeley, and I decided, entrepreneurship was not for me, and moved on to become a faculty.
At UCSC.
And before that, I was in Keck, and then later on I moved to
Keck
UCSC.
USC?
Yes, Keck in Claremont
Oh,
College.
Claremont, not USC.
Yeah.
Or fight
Well,
on
I mean,
USC.
they're all funded by the same.
So,
Yeah.
What drugs were you successfully able to deliver with that? And what is Therapeutic doing right now, Bio?
So I think BioAura is doing 2 blockbuster drugs. One of them Humira, which is really interesting. So because my dad and a lot
What
of
is
people
that?
around these are oligo-based drugs for chronic diseases. And for example, Humira is sort of a anti-TNF alpha based drug, which is sort of when your immune response gets activated too much, you want to suppress it. And that's the job of Humira. Humira is for diseases that are a result of overactivation of your own immune response that is triggered by environmental factors or other factors. So a lot of people who have Crohn or IBD disease, these are diseases of the GI tract, small intestine, where they get a lot of inflammation there.
Yeah.
And it's a very it's not a good disease, because you constantly have to get injection. And the problem is, because it's in your gut, if you're taking an injection, very small amount of the drug gets to actually your gut. If you can deliver it locally,
Easy.
you're reducing the required dose for those. So I think these are the 2 drugs that BioAura is trying on. What I did, which actually made a big paper in Science at the time, I tried to do vaccination with it, because that was an easier thing. Because vaccination, what you measure when you want to measure vaccination, you introduce a protein to the body, and then you look at that is not endogenous, meaning it's a protein of something else, a chicken or a mice or something else. And then you look at the immune response and see how the immune response is reacting or responding to that pathogen. And this is basically vaccination. When you get vaccinated, you put a pathogen or a protein from what you see as flu vaccine
Right.
is a combination of like proteins or pieces of cell membrane that contains that protein. Then your body started creating an immune response. So then you become protected because the amount that we deliver is very small amount. It's not enough to make you sick, but it's enough to,
create a response.
Exactly. So this is what we tested. And what we also realized, this is a really good question because sometimes when you invent something, you actually don't know where the application would be until you start doing it. One of the major power of this technology, the oral technology, which I think someone will license it for vaccination, is that a lot of vaccination are still injection.
Right.
Because it's easy. Again, the same vaccines are protein-based.
More efficient, right?
Vaccines are protein-based, so you can take them orally. Why? Because the
enzymes.
Yes.
We get back to enzymes.
Exactly. So that's why we give them by injection. And then you create a lot of systemic immunity, we call it. That means that there's a lot of antibodies will be produced in your blood. So if next time the same, pathogen from specific to a disease gets to your blood, then you have this antibody circulating. They can capture it and destroy it. But remember, a lot of our infection, or let's say respiratory infection, like COVID,
right?
They're not introduced to your blood. They're first introduced to your nasal cavity or breathing, or some of them are oral, like cholera. So you don't want to wait until the pathogen gets to your system before you can,
block it.
Our body has another type of immune response. We call it IgA. So the antibodies that are produced in your blood, we call them IgG. And when we measure something in the blood, we measure IgG titer, we call it. But when something is, there's another immune response we call IgA. That's in your mucosal tissue, in your mouth, in your gut, in your basically whole GI tract that are produced. If we can activate those, if we can generate IgA for a lot of respiratory diseases or oral diseases, you can actually take drugs orally and create IgA. So now you have the first line of defense in your gut. But we have never been able to accomplish that because, again, proteins are broken by enzymes, so we have to give them directly to the bloodstream. When we did that study, we not only identified like some of the drugs were getting to systemic circulation, we identified the whole immune response, oral immune response to it. So that means that there was a potential for the technology to be used for oral vaccination rather than injection. But not only that, you're not only getting the benefit of systemic vaccination or injection, but you're also getting oral immunity now. That means that you're actually combating these diseases
on two fronts.
At their entry level, and if they get to the bloodstream, you have another second line of defense. So I'm very hopeful that I don't want to deal with vaccines because the vaccine commercialization of vaccine is very difficult. It's a market that is,
and there's so much stigma, for the lack of a better word, around it.
Yeah.
Why is that?
I think vaccines are difficult
People don't like it.
I mean, not only that, like if we're talking about the marketing of vaccines, the major people who are investing in vaccine technologies are mostly the Gates Foundation. Otherwise, I think that the U.S. government and those funding agencies, we are very reactive. We're not responding to problems. We're reacting to problems. For example, the mRNA vaccines, we had those from years and years ago, but no one wanted to invest in them enough, for us to be ready for when the, COVID hit. If we invested enough money in those technologies, looking into potential risk in the future, we were not, we could have had the vaccine much faster, I think. So I think, but people forget. So after the COVID, there was a lot of conversation about now we're going to look forward, we're going to see what technologies we need. But once the COVID problem, faded, those promises also faded with COVID. So I think we need to develop a better strategy of people who are, investing in future technologies. These should be people who are risk taker. I think we should bring business people in those decision-making processes, because sometimes I feel like business people can play a better role in preparing for future, because we were always prepared for future. So I think this is why not many people we see them moving to vaccine industry.
The other issue again, I see and I hear is it's almost like a one size fit all. The dose is fixed. The formula is fixed. No one is studying my body to see, okay, what is it that I actually need?
Yeah.
Do I really need, what's the response of my body to, it can't be all the same, right? It can't be all bodies respond similarly to the vaccine. I think that's another thing when I talk to people, it turns people off from using vaccines. Like here, kids, adults, they're all getting the same formula. It doesn't make sense.
Yeah, I think, no, your point is well taken. I do agree that, our immune response is very different. My body could, react to much smaller dose than someone else's body. Having said that, when you talk about something at this massive scale, it's very difficult to do those sort of things, right? You, of course, can create technology. I can develop a technology for you today that if you get vaccinated, you can measure how much, antibodies you're producing in the next 14 days. But you have to also think about it. How many people will have access to this technology? So what it'll create, I think it'll create more segregation between the rich society and the poor society that some people have more access or less access to technologies. Having said that, in future, if technologies like my technologies are scaled, these are the technologies that can really, be produced at a cheap price where you can measure your antibody titers.
Yeah, exactly. We haven't got to this yet, but I hear you in that. What happened to the insulin? Were you able to deliver insulin?
No, so I think there are other people who are still trying to deliver insulin orally, and the challenge with insulin, as I mentioned to you, is that the very narrow therapeutic window. Because with some drugs, if you deliver a bit more, it doesn't harm you that much. You might get a bit of side effect. With insulin, you can put people's life in danger. So that's why they're still trying to figure out what is that narrow therapeutic window, which is very different from patient to patient. So I think that's what makes it a bit difficult for oral insulin. There are some sort of formulation, but for type 2 diabetes, not type 1. So I think it's different.
Too much of it, I hear you, but you always get too little of it with this method. Is that right?
So
can you deliver? You're not going to deliver more than what you need because that amount is fixed.
Yes, but
it's going to be too less of it.
Yeah, if you deliver less of it, then it might not be effective. So you have a very narrow window. So you always have to be within that window to be able to have an effect. Because with insulin, you also need the insulin to be absorbed quickly because this patient will need it right away. It's not like something that you can wait. With Humira and other drugs is different because. Right. You take it a bit here.
You can't wait. I see. You can't wait. I want to go back to those enzymes, and it makes more sense when in the drug problem. So is your research currently at UCSD around controlling the enzymes or sensing what's happening with them?
Both. So I constantly have my eyes on what comes out in the industry. And I have created some sort of a connection with the industries who are, in the business of scaling up these enzymes and making them more stable and whatnot. But I'm really looking at enzymes that have a function that could be beneficial for us. So if you want to go, we can dive into the CRISPR-Chip idea.
Yeah, actually right timing then.
Perfect.
What is CRISPR?
So CRISPR is this enzyme that was, or this molecule. It's basically a molecule composed of two things. One is a nuclease enzyme. Nuclease enzyme is an enzyme that cuts nucleotide. And then there is a GPS made of RNA attached to it that actually tells this cutting enzyme, the scissor, where to go and cut.
Can you break it down less complicated? Which one is the scissor?
The enzyme, the Cas9 we call it, is an enzyme. And then there is a guide RNA, which is the GPS of that enzyme, tells the enzyme where to go. Yes. Together, we call the technology CRISPR.
Okay,
these two combinations.
What does it do?
So first it was discovered in bacteria, actually. So some scientists, they were looking at why some of the cheese that are produced, it comes out well, and it's forming well, and then some type of cheese, it goes bad. Then they identified the cheeses that actually don't go bad in their DNA sequence have a code, a very specific code that sort of assembles the viruses that were attacking them, which was really interesting. And the cheese that went bad did not have that. So they hypothesized that there is something with this, bacteria, because making cheese all, putting different type of bacteria in them and letting them, make the cheese for you. These bacteria have some sort of an immune response against those viruses that is coded in their DNA. And the cheese that went bad, those bacteria did not have the code in their DNA that was protecting them against those viruses. So this was how the whole field started, and many scientists started to look to see what those DNA code, actually what enzyme they encode for. So then they found this Cas9 enzyme, and the guide RNA that is attached to Cas9 was exactly resembling the virus's DNA. Many years later, the whole mechanism was identified by Jennifer Doudna and Emmanuel Charpentier. So Jennifer Doudna was in Berkeley, so I was constantly exposed to all of the hype
amazing.
to Berkeley,
What year is it? 2016?
2013, 2014. This is where the hype was really high in Berkeley. And I was doing mostly I was working on the capsule, I was designing other diagnostic, but I was just fascinated by this whole technology. So I remember one day it was 6 a.m. I was going to bar method, not bar, it's bar method was an exercise class. And on the way, I decided to stop by and have my coffee because I had some time. And I was standing there outside, and my whole mind was just fascinated by CRISPR. And then I thought, this is a great, robot. It's one of these robots that can unzip the double-stranded DNA, look for its target sequence, identify the target sequence, and then cleave it, cut it. So this is the mechanism that bacteria actually use to kill viruses.
Right.
So these CRISPR complexes, they float in the plasma or in the cytoplasm of the bacteria, and then once they see a virus that they can recognize, they chop the virus off and destroy the virus. So we humans learn to use that cutting, that capacity to edit the genome. So what we do, we find the target in the genome, in human genome, and we can direct the CRISPR to go and cut that part, the specific part, and then we insert the
whatever we want to put. And then what does CRISPR do?
So the CRISPR first, the DNA is a double helix, right? So, and it's very convoluted. So the enzyme first goes, and you see the enzyme hopping. The enzyme, it doesn't opens the unzip the whole thing. It hops on the DNA first because on the sequence, and then all of a sudden you see it stick to a part, and that sticking part is because there is a sequence on the guide RNA, the GPS, that recognizes very specific region in the genome. Once it recognizes the specific region, then it unzips it, and then it tries to match the GPS to the sequence. Does it match? Doesn't match? If it doesn't match, it hops to the next one. That's how it functions. If it finds the right match, then the enzyme sort of changes conformations, turns around and it has a scissor part where it cuts the DNA. It cuts the double-stranded DNA.
And the RNA is what it inserts?
The RNA is basically finding the target region.
I see. That's the GPS.
Yes. The insertion process, it's a whole different process.
That's biological process, right?
Yes, there is a biological process that the cell, because your DNA is constantly randomly cut, when you do an X-ray,
right.
You're randomly chopping your DNA, but then the cell has a process that can fix the DNA. So we can allow the cell to fix it and hope that the cell does a good job in fixing the genome and bringing it, making it the right genome. Or we can deliver a template, we call it, that has the right sequence in it. And then we have chemical method to insert that in the DNA. So you can imagine the potential are huge, because if we identify someone with a genetic disease, like sickle cell disease, Duchenne muscular dystrophy, these are diseases that the patients are born with them, and they're really debilitating diseases. So that now that we have found a cure for it, not cure, a therapy, we can actually go. It could be a cure. So you can go to the DNA and cut those regions that have a mismatching code, or the code that is making the person sick, You cut that, and then you insert the right one. Not only that, if you want to be a superhuman, if you want to have like some sort of be tall or have blue eyes, we know the codes for those, phenotype, we call them, those characteristics, we call them, right? So we can go and cut that.
Even if you're like an adult, you can change the color of your eyes?
Well, so for those things to be effective, I don't think like you potentially can do it. I don't know. Like now we have people are reporting it that they're fixing hearing loss.
Wow.
By genetic hearing loss by this. So I think you can engineer human in the future, where you can actually just basically the same way that you print your material, you can print your
height
DNA
and weight.
Yes, yeah. And I want the height to be this, the eye color to be this, the muscle mass to be this. So there's a lot of ethical
conversations, I'm sure,
around this. So I'm not doing any of that. What I'm doing, I'm actually just using the enzyme for what it does. And it's basically a search function. Why is that important? What does it do for me if I use that? You may ask.
Search function, as in,
so that the CRISPR molecule, as it finds a target sequence, what does that do for me if I wanted to harness that power?
This is back in Berkeley,
yes.
Or you're talking about now? Okay, back in
Berkeley, yeah. I'm like, okay, if I can program this CRISPR molecule to find very specific region in the genome, I can potentially detect diseases using that sequence. And why do I want to combine CRISPR with my electronic platform? It's important to know that everything in our body is electronically charged. Our DNA is highly negatively charged. Our proteins are charged. These are charged molecules. When we talk about something binding to something in the body, it's this electrostatic interaction. People don't think about it that way because we're not used to thinking about it this way.
Hard to imagine it.
Yeah, but coming from an electrical background, which is now coming very handy for me, I really take a different perspective, different angle at looking at science. To me, everything is electronic. So, cell-cell communication is electronic. The way tissues are performing or functioning is electronic to me. So if you ask my students in my classes, they hate me because I teach heart and vascular system all with, drawing electrical circuit and models. But anyways, so to me, I'm like, okay, if I combined the CRISPR enzyme that I programmed to detect a specific sequence in the DNA with my electronic transistor, which are designed to detect electrical signals, every time this CRISPR molecule hops on something, binds to something, or cleaves something, I can detect those electrical interactions. And this is what we did. So we anchored CRISPR to our transistors, which you may ask me, what is a transistor? Probably now, so I can explain.
What is a transistor?
So everything in your computer, in your cell phone, your CPUs, your GPUs, they're all made of this little component called transistors. And transistors are basically an electronic circuit that can detect electrical signal, amplify electrical signals, and this process that we have in our electronic gadget, it's all, the foundation of all of them is transistors. You may want to think about it as 3 electrodes. You have in transistor, you have source and drain, where source is creating the source of your electron. It is passing through a material that you have between your source and drain. Drain is where the electrons are drained. And then specifically in transistor, we have the 3rd electrode or 3rd terminal that controls this field. And it's important for our application. I don't want to get into that. It's very technical, if you allow me to not dive into that, because I don't think I can explain that easy. But so for my research, I was using a lot of transistors for monitoring biological interaction. And I was working on a very special type of transistor. These are quantum transistors. They're not like silicon transistors. They're very different. And I'll tell you why quantum, what type of a quantum material we used later. But yeah, the whole idea was that can we actually monitor the CRISPR enzyme as it binds to its target compared to when it doesn't find its target?
Wow.
Because then you can imagine having a chip like that where you can take a swab or the nasal swab that you take for COVID, put it in a solution, add a drop of solution on this chip, and the chip can tell you if you have certain diseases or not. And it has a lot of application for resource poor setting where we don't have the facilities, the instrument to do this type of testing. Because right now, the way genetic testings are done, they take your DNA sample, which could be a buccal swab or DNA sample from anywhere. Then we chop the DNA. Then we look for our specific sequence that we're looking for a specific disease, because we know those sequences. Then we have to amplify. After we amplify, then we label with optical reagent. We label, and then we read those labels. So it's a very tedious process. It requires equipment, instruments. So there's a lot of effort into how do we make this more accessible to, for different people, different applications. But then this chip can do everything.
This chip that I'm holding in my hand
Yeah.
can read my DNA, sense the process of clipping the DNA and programming it.
Yes.
So this is the gateway to digitize the body.
Yes, it's a gateway to
Wow.
biology, right? And CRISPR is not the only enzyme that we can monitor its function for a specific application. We have another project with the Gates Foundation where we use the olfactory receptors from insects.
Right.
So I talked about, the CRISPR enzyme, we're using it from bacteria.
Right.
We're still trying to identify, do we in our body have some sort of a CRISPR-like enzyme which we can use? And that has a lot of application for gene editing specifically. But as you see, when I talked about harnessing the power of biology, beyond our, human organism, there's a lot of other organisms that we can use. So for CRISPR-Chip, we use the CRISPR from bacteria. For the project with the Gates Foundation, we looked at insects. So, insects and dog, they can smell much better than us. And the reason they can do that, because they have a very high olfactory receptor sensing capacity. Many receptors in the nasal cavity that they can sort of capture the odorant, different smells. And there are channels that are opening and creating some sort of electrical response that gets sent to your neurons in the brain, and then you can sense something. We wanted to see if we can create an electronic nose that is much better than us in sensing and smelling, because we human are not, but we don't have the strongest sense of a smell. Can we actually harness the power of this organism and create something that could potentially help us?
So like amplify our smelling properties?
Yes.
That's amazing.
Yeah. So, and the project was that, can we actually smell diseases? Because a lot of
animals
can smell diseases, right? So we collaborated with scientists from Rockefeller University in New York, and a startup in France who was, they were sort of expert in mass producing this type of olfactory receptors. Olfactory receptors are very specific proteins. We call them transmembrane proteins. They sit on the cell membrane, and they have a capacity to open and close ion channels.
I see.
Right? So it's like, imagine a tetramer that is closed, and then once it binds to an odorant, the tetramer opens up, and then it allows things to pass through, and it's like a signal that gets sensed by the neuron then. So that's what we did. So the Rockefeller folks, they identified a couple of olfactory receptors that were a general responder to a range of different odorants, but different signal. For example, they were smelling glucose, but then they would give a different signal to glucose than other things. And we mask scaled it, and then we combined it with our technology. We were able to, with a single olfactory receptor, a single protein, we were able to sense over 30 different odorants and distinguish between them.
That's amazing.
Using the chip. And then
this is still at Berkeley? Or
no, these are back as a faculty.
Berkeley.
Yeah. We just concluded that project.
That's amazing.
And you can imagine a day where you can have not many, like 4 or 5 proteins that are general protein olfactory receptors from dogs or insect, where we can mass produce with synthetic biology. And then we can look at how these olfactory receptors are responding to different odorants, and then use machine learning to identify their correlation with different smells.
That's right.
And you can have a brain that basically can decode the different smells and odors for you, from disease to like the smell of a vanilla or a, I don't know, some of
Yeah,
the...
different symptoms identified, correlated. That's brilliant. So CRISPR-Chip, that was in Cardea. Later became Cardea. What was that story? Because
Yes.
you're fascinating. That you're a scientist, amazing researcher, and a great entrepreneur.
Yeah.
I want to hear the process of Cardea.
Right. So when I invented CRISPR-Chip, and I wasn't actually planning to commercialize it at all.
Uh-huh.
It was just cool. I was happy. I wanted to publish it in Nature, which we did. Then around the same time, there were a couple of other companies rising, like Mammoth Diagnostics or Sherlock, which were also based on CRISPR. They were using CRISPR for diagnostic application, very similar to what I was doing, but they had no electrical platform. They were mostly optical platform. So they were really the driver for me to
Inspired you.
Yeah. So they and I met with one of the co-founders of Mammoth Diagnostic at the time. And I realized, well, I had enough data that I could actually pursue that path again. So this is what I did. I started looking for to see who would be interested to partner with me, and then I found my co-founders. That they were sort of scaling up the chip itself, and I was becoming an expert in combining biology with electronic, and understanding what are the interfaces that was needed. How do we read the signal? How do we process the signal? But I actually hated making the chip itself. Going back to my undergrad, where I want to see the impact. So the process was sort of boring for me, and I just want to get to application. But one of my co-founder and him and the other, one of his other friends, they started a company prior to me starting, Cardea, to just scale the chip for various applications. So I partnered with them, and this partnership have been the most amazing things in my life, because it's a friendship that that has grown to designing many more things together. And at some point, we felt like there was no point of working separately, and we combined and we create Cardea Bio.
And you won the National Academy of Inventor Award, or a fellow now.
Yes. So
when did that happen? And that's because of your invention in the CRISPR-Chip?
Yeah. So I have about 32 patents, and CRISPR-Chip, the Mucojet, the Biojet that you saw, and a couple of other technologies were sort of all the result of my academic research that we commercialized. And National Academy of Inventors is really looking for academics who are leaving an impact with their research. And I think I was selected this year as a senior member. So
that's a big deal. There's only a couple of hundred of them, right?
Yes, yeah. So we'll see.
Very good, very good. So what are you working on today in your research?
So CRISPR-Chip was...
you sold that company,
that company was sold, but then there was a version. It's the same technology, but we were doing other application with it. That we had to spun up another company from Cardea called CRISPR-QC. Mm-hmm. What CRISPR-QC does, it's not genotyping. It's sort of using the exact same platform, but looking at the function of CRISPR in real time, looking at its kinetic, how fast it's cutting the DNA, how efficient it's cutting the DNA. Because it turns out that people who are doing gene editing for treating those diseases, they really needed a platform that can reduce the number of the formulation that they were gonna go test in cells. Mm-hmm. The reason that CRISPR therapy is very expensive today is because you have hundreds of formulations that you don't really know which one will work better for that specific patient. So the process is that you take all of those formulations and then you test them all to find something that works, which is not very easy. It's very time consuming. It's very costly. So after the first FDA approval on CRISPR editing, which was 7-8 months ago, and for sickle cell disease, now the game has changed for these companies. It's not now about the race to who gets FDA approval anymore. it's about who can make this technology more scalable. The price becomes more important. The safety becomes more important. So now there's really,
Wow.
so that's where, CRISPR-Q sits in a nice place where we're trying to really offer the market an analyzer that can analyze the quality of their formulation to allow them to reduce costs and increase the speed.
Get to market faster.
And get to market faster. So that's on the company side, because CRISPR-Chip has still, potential to, result in more technologies. But on the academic side, I am very much involved in, so there are other curious questions in my mind. So again, I like combining biology with electronics. So what I do right now, I'm making these chips at a very much smaller scale. So this chip has transistors inside that are about 10 micron size. And micron size is small enough, but I want to make them even smaller. I want to get to nano size, and we have produced nano size transistors. And why do we do that? I want to get to single molecule monitoring, meaning that I want to see how a molecule actually changes in real time. So that's one project that we're doing. It has application, but I think it's we have to talk for another hour if you want to dive into that. But the other part that I'm also interested in is how do we actually bring more electronic to make more autonomous system for testing. And the reason I'm saying that is that right now there is a huge market and field in removing the use of animals from testing and moving to chip organs. Basically, these are organs that are fabricated and printed on chips. So we can do drug testing, we can identify the growth of like stem cells in different conditions. There's a lot of space application for them. And what I do right now, I sort of make these organs like brain, bone marrow, and then we have integrated electronic because we want to remove human from every aspect of measurement, because us humans are the biggest problems when it comes, to testing. We create a lot of variability. And it makes the testing also more hectic because someone has to constantly test something or put probes to measure something. So these are integrated electronic organ chips that can monitor the passage of drug across the brain, the impact of different formulation to the integrity of the brain, stem cell differentiation, things like that. And if you may ask me, why are you doing all of these things? Now I'm interested in understanding aging. I want to see how do we age, why do we age, and what happens to our blood when we age, and can I use my engineering techniques to identify some of those mysteries and help biologists to sort of, and drug developers to sort of combat aging.
And what kind of research have you done in that aging and longevity so far?
So again, I told you, a lot of my invention is a result of a curious question that I needed an answer for. And really, being an academic, it's an amazing job because you just wake up in the morning and you're like,
why?
I want to do this. Yeah. Why is this happening? So let me just, figure it out. So back in Berkeley, I was also involved in another type of research because, as a postdoc, you have multiple different research areas. We were exchanging blood between young and old animals. And then we were looking at the rejuvenation effect of the young blood on the old animal, which was an amazing topic. My colleague, Dr. Irina Comboy in UC Berkeley, she's an expert, renowned expert in that. But one of the things that caught my eye is that after a single blood exchange between young and old animal, the young animal brain suffered. When I say suffered, that means those mice were losing their capability of like some testing that we were doing to examine their brain function. And then I thought, okay, a single blood exchange, what is that like significant impact? Where is it coming from?
Yeah, what did it happen?
So when you think about the blood exchange, you're exchanging blood between two animals. So the blood has two types: is the soluble part, which is the plasma, and then there's the cell types, and which are white blood cells, red blood cells, platelets. What are the most abundant cells that could result in a major impact? These are red blood cells. And then I talked with my PI and I realized no one is actually looking at red blood cells because red blood cells are considered just the oxygen carrier in our body. And I couldn't just it was in my mind the whole time. So when I started my faculty position, I decided to actually identify to see, is it actually only oxygen that is the role of these red blood cells? So what we identified was that actually as we age, our red blood cells become more stiff. They lose their capacity to deform.
Mm-hmm.
So what happened when a red blood cells passes to your vessels or your brain vessels, capillaries? They're about 5 micron, and they sort of deform and squeeze and easily pass through the capillaries. As we get older, they become more stiff. So now as they pass through the capillaries, they experience a lot of stress, a lot of force.
Mm-hmm.
Then we realized that, okay, the force on red blood cells creates nitric oxide. There's a protein on the red blood cells called nitric oxide synthase that produces the nitric oxide if it experiences shear force. And so one of my PhD students, it was one of my PhD students' project where he started identifying, taking old red blood cells, young red blood cells, exposing it to physiological shear and forces, and seeing what was happening with this. And we identified that, yes, as we get older, our blood-brain barrier, that's the vessels between our brains and the blood, it gets more leaky when we expose it to old blood.
Oh.
So this opened up.
That's why the animal suffered the...
Yes, so then you're like, okay, so what happens when the blood-brain barrier becomes leaky and opens up? That means a lot of things from the blood that were not allowed to get to the brain, now they're getting to the brain. And when you think about it, when you take an MRI of an old patient, you see heme molecule in their brain when you don't see that in the young animal. So these are the heme molecule, it's coming from the red blood cells. So that means that some of these red blood cells content are getting to our brain. So then the next question came about, which was a thesis of my second PhD student, who she was working on identifying, okay, what else goes from the inside of the red blood cells to the brain?
Why the brain barrier becomes leaky? That's as a result of red cells become stiff?
Yeah, so when the red blood cells become stiff, they produce nitric oxide. So
nitric oxide make it...
Nitric oxide we all know it as a vasodilator, so it basically dilates the vessels.
So our body keep producing red blood cells. What happens that the new ones that are generated are also stiff? Because yes. I can imagine the new ones got to be intact.
Very good question. So yes, the red blood cell life cycle is about 3 months. When we talk about old and young red blood cells, we're not talking about the newly generated red blood cells. We're talking about the stem cells that are in charge of producing. Because the stem cells in your bone marrow, they produce your red blood cells, your white blood cells, like immune cells. This is what the cells in your bone marrow does. As we get older, something happens with the bone marrow that started producing different type. Yeah, so it doesn't matter how often they produce it. The things that they produce are not as high quality.
The factory become rusty.
Yes, exactly. But then there is potential to it because if we can identify what is it that is changing in a stem cell, there is therapy for it.
Yeah.
The stem cell therapy, bone marrow therapy. And this is why I got super excited about this research, And we also identified that within the red blood cells of old compared to young, there are biomarkers, there are RNA molecules. And I'll tell you what RNA molecules are. But these microRNA that we identify, some of them were associated with neurodegenerative diseases like, Alzheimer, Parkinson. So there is a signal that these red blood cells carry with them as they get old that is associated with these diseases. And it might be why we saw those brain decline in the young mouse, in Berkeley. So it's still developing this project. It's developing, super excited about it. And, we want to identify to see at what stage red blood cells start packaging this biomarkers or these signals to them that could be potentially the cause for neurodegenerative diseases.
What was it? Nitrite? What is it?
Nitric oxide.
Nitric oxide. And how do we measure it in your blood?
So nitric oxide is it's a very short-lived molecule. Mm-hmm. So it just gets, it's a reactive oxygen species. It just gets produced and destroyed, but it leaves its impact. So it's hard to measure it. But then we have mechanism and assays that we can measure it, immediately. So there are like an optical assay that some labels that you put, and then you see the labels turn on as a result of the nitric oxide production. So what we did, we actually measured to see how much nitric oxide the old red blood cells were producing compared to young red blood cells in presence of physiological shear stress, physiological forces. Because if there is no forces, they behave the same. Under forces is that when you see things are starting to be produced. And then when we tested them in our organ-on-a-chip systems, we realized that, yes, they're creating a significant leakiness in the brain. And that leakiness, we were measuring it electronically.
Sure. What can be done in the dental world? I know you haven't been exposed to it, and I tried to inspire you.
Yes.
What is it that you think can be done?
Well, can we not go to dentists anymore?
I don't think if our audience will like that, but...
well, I'm interested to know, and we were just talking about it, I'm interested to know what happens to our teeth as a result of environment. And when I talk environment, if you're exposed to space for a long period of time, we have significant bone loss. And bones are sort of regenerated because they want that force, that gravity, it impacts the bone regeneration. How about the teeth? Does something happen? I mean, have you guys looked at astronauts to see if astronauts have worse teeth when they come back or no?
I'm sure someone has.
Yes, but I think those are cool to look at and see. Also, technologies that are developed in different sector, in commercial or in for different applications, sometimes can find use in different field. For example, when we talk about testing and diagnostics in space, and we put so much funding in it, it's a resource poor setting, space. You don't have access to many things. You have to have, you need to make sure that you don't need like cold storage, things like that. But the same exact technologies are needed in Africa or in resource poor setting. Why not like synergize the field to see who's making what and how can we bring these technologies together? For example, 3D printing, it's becoming a thing in a space because if we go to Mars, we have to 3D print things.
Sure.
But can we carry, huge 3D printer with us? Of course not. Every gram of things that are sent, to ISS now, it's measured very accurately. So you can't really, the cost of sending things is significantly more than the cost of building things. So putting an expensive 3D printer in Mars is much better than sending, a lot of cheap printers to space. So, or even in ISS, where now we're doing a lot of experiment. Can we actually start using 3D printing things like organ chips? We're starting to use a lot of organ chips in a space to measure how the environment, the zero gravity, the radiation impacts the cell viability, toxicity, the stem cell regrowth. Can we actually, print those tissues in space? For that, you need it to be compact, easy to use, not have a lot of reagent to be sent with them. So, I think there's a lot of synergy between different industries if you think about it.
Very exciting. And another idea, in the field of ortho, back to the robot and controlling the robot. Body is a robot. We want to control it. What happens that we grow crooked teeth? And if you can biologically control that, we can potentially put orthodontics out of the business. But overall, that could be another field that, I don't know, I'm sure there are answers to that. I haven't researched that.
No, that's interesting. I think that comes from looking at factors that actually shape our gum structures or things like that, and how are they designed, which could potentially be a genetic code for that, right?
That's what I'm thinking.
Yeah, we can identify and basically edit it.
Instead of putting braces, you take a pill,
yeah.
and boom, all of a sudden you have straight teeth.
And also
grow straight.
I think there's a lot of genetic factor to it. So maybe we genetically modify people so they don't need dentists anymore.
It is definitely genetic, the same thing, yeah.
So maybe
I think we found our project together.
Yes, maybe dentistry is
amazing.
Also becomes a gene editing.
Thank you, Kiana, for coming to the podcast.
Absolutely.
Very exciting field you're doing, and all the best of lucks.
Thank you.